and you can have everybody here. All right, so I think we should get started.
I'm on your time.
All right, so let's try to keep the seminar schedule. All right, so let's get started. First, welcome everybody to our first seminar for this semester, the STEAM H seminar series. Well, again, as I said earlier, so there would be people joining in at any time, probably right now. Okay, all right, let's continue. Again, welcome everybody. I know each of you might have a lot of work to do, so I appreciate you being here. This is our first seminar for this semester's STEAM H seminar series, right? And you might be hearing a ding ding sound. I'm trying to do the introduction while I'm meeting people into the seminar. This semester we have 9 seminars scheduled, right? So we have a very good lineup of speakers. We have a distinguished speaker to line up, including our today's speaker, Dr. Griffith, which I will introduce in a minute. So I hope to see all of you in the other seminars throughout this semester. Again, all of you have registered, so that registration is good for joining all the other seminars throughout this semester. So you don't need to register again. So just keep that confirmation email you have received. Right? Okay, with that said, let me introduce today's talk, right? So I cannot think of any other speaker, any better speaker to start our seminar series today. Doug Griffith is a well-known evolution biologist. He's also a pioneer for African-American scholars in the field of evolution biology. So I believe if you are not the first, you were the second person who ever received a doctoral degree in evolution biology, right? African-American, okay. Dr. Griffith graduated from Auburn College, and I believe in 2017, he was named the outstanding graduate of biology at Auburn College. Is that right? Give me a second. Some other people are trying to join in as well. Okay. And he got his PhD. He received his PhD in environmental, evolutionary, and systematic biology from Wayne State University in 1988. Right. So in 1994, he was elected a fellow of the Council of American Association of the Advancement of Science, AAAS, right? Distinguished honor. And he has been working at North Carolina A&T University for 14 years. And right now, he is a professor of biological sciences, but he was associate dean of Zhong School of Nanoscience and Nanoengineering at North Carolina A&T. And he at North Carolina A&T, he has been the director for more than two actually, student education and research program, including a graduate IBM program that's for graduate program in bioinformatics, right? And he was also the director for the Beacon program supported by NSF, I believe. Probably he will talk a little bit about those two programs, or at least acknowledge the two programs that he will talk. And again, as I said, he is a well-known evolution biologist. He has written prolifically a few books, and as I was told just before the seminar, he is working on his third book. And he finished 2 books in the last year. Congratulations, Doug Griffith. And again, he really is well known in many aspects of evolutionary study, including the study of human races. So without further ado, I will give the floor to Dr. Griff, who will talk about his most recent research supported by one of his grants. All right, the floor is yours, Dr. Griff.
I'm going to share my screen, and I want to, okay, yeah, okay, so there we go, yeah. So I'm going to share my screen, and I want to thank you all for inviting me to your seminar series. Looking at the list of people who are going to be speaking for you this semester, I am greatly honored to be included in such an esteemed list of scholars. So what I'm going to talk to you about today is work that was funded off of NSF grant out of the Nanobiosciences Directorate that studies the impact of nanomaterials upon living things. And for those of you that don't know my history, actually, my early years were in Drosophila evolutionary genetics. And so I did experimental evolution on life history traits. But when I came to the Joint School of Nanoscience and Nanoengineering, really couldn't run a Drosophila laboratory in a building with clean rooms. So I had to find a new avenue of research to do that was one relevant to nanoscience and nanoengineering, and also wouldn't get all the rest of the faculty really mad at me. So I decided to start looking at the effect of nanomaterials on bacteria. So, and in that time, this is a picture of my genomics research group that I built at North Carolina A&T. Now, this group, this is a picture from us at the 2018 Congress of Biocomputational Evolution in Action, which was a National Science Foundation Science and Technology Center funded between Michigan State University, University of Texas at Austin, the University of Washington, the University of Idaho, and ourselves. But the principle of my research group is to allow for tiered mentorship, starting with me, a senior researcher, but also working with associate professors, assistant professors, postdoctoral researchers, graduate students, and undergraduate students. And you see all of those in this picture, many of whom contributed to the research that you're about to see today. Assuming I can, yeah. So now, for this study, I'm going to talk about the chief collaborators were Dr. Misty Thomas, Dr. Jessica Hahn, Dr. Scott Harrison in the biology department at North Carolina A&T, Dr. Judah Wonkum, who was my postdoctoral researcher, and he's now appointed as a nanoscience researcher at UNC Greensboro, Dr. Kristen Reinhardt, who was then a graduate student, but now is a faculty member in the computational data science program, undergraduate Anna Tapia, my former graduate student, Sade Boyd, who is now Dr. Sade Boyd, and she's a postdoctoral researcher at UCLA, and the undergraduates, all of whom have graduated, Emma Van Beveren and Bobby Yang. Now, this work, by the way, contributed to my capacity to write this book, which is shown at the bottom of this slide, which is going to be coming out later this year, called Antimicrobial Nanomaterials, Principles and Applications from Elsevier. And what I do in this book, which is different from what most of the work that has been attempting to apply nanomaterials as antimicrobials, is that I put the use of these materials in the context of microbial evolution. And so one of the things that led me to the experiment I'm describing today is the fact that people are attempting to apply these antimicrobial nanomaterials with no cognizance whatsoever of how microbes respond to novel materials in their environment. So, and the reason this is such a fallacious worldview, okay, is because microbes, as you all well know, have been exposed to toxic materials, including metals, since the beginning of life on this planet. So the idea that virtually all bacteria have genes to resist toxic metal ions such as silver, cadmium, mercury, nickel, zinc shouldn't be surprising to anyone. Now, the largest group of resistance mechanisms involve energy-dependent efflux. And we found this, that adaptation to silver resistance, both nanoparticle and ionic silver in E. coli, was mediated primarily by mutations in the Cus-S gene, which is part of the Cus-S, Cus-R two-component response system that then initiates the formation of efflux pumps that move silver out of the periplasm of E. coli. Now, next in line are enzymatic transformations, oxidation, reduction, methylation, demethylation, or metal binding proteins such as methylthionein, chaperone, COP-Z, or SIL-E. In addition, bacterial clones may slow their growth or cease dividing and enter persister states in the presence of toxic materials. Now, on the other hand, there are good metals that are required for growth, such as iron, sulfur, manganese, magnesium, and so on. Now, of these, acquiring iron is the greatest challenge for all bacteria, and I would argue for all life. And iron predominantly occurs as ferric iron under aerobic conditions. But ferric hydroxide is poorly soluble in aqueous solution. So under anaerobic conditions, the equilibrium shifts to ferrous iron, iron 2, and that is more easily available to microorganisms. Now, as you all know, E. coli is a facultative aerobe, so it can operate both aerobically and anaerobically. Now, iron availability is known to be a key to pathogenesis for a variety of microbes. Thus, many innate immunity mechanisms utilize iron sequestration as a way to control the growth of toxic parasitic organisms such as bacteria. Now, as iron is so important, microbes evolved means to take it up from the environment, such as the siderophores like Enterobacter. Now, this siderophore in E. coli is synthesized by genes such as ENT and FEP, and their expression is controlled by the global iron homeostasis regulator, FERB. Now, excess iron is known to cause oxidative damage up to the point of cell death. Now, since iron nanoparticles are being proposed as a method to control multi-drug resistant bacteria, we wanted to ask the question, can and how does E. coli evolve resistance to excess iron? And we've now published a series of papers on this question. The one that I'm going to be talking about today is Thomas et al. Too Much of a Good Thing, Adaptation to Iron 2 Intoxication in E. coli, which was recently accepted in Evolution Medicine and Public Health and is being published this year. Now, for those of you unfamiliar with the toxicity mechanisms of silver and iron, they are known to generate reactive oxygen species, or ROS. Both can bind to thiol groups, although silver does it better than iron. They both interfere with transcription and translation. They both interfere with the cell wall and the cell membrane, often leading to the release of cell components, which is at the bottom. And they both, excess amounts of iron and silver, both interfere with the respiratory chain in microbes. And of course, I like using this joke. If Iron Man and the Silver Surfer were to team up, they would be alloys. Now, the mechanism of iron homeostasis are ancient. And we could ask the question, how do bacteria evolve resistance to an excess of an essential micronutrient? Now, this is a problem, right? If iron is the most crucial micronutrient for bacterial growth, would think that virtually every bacterium has a very powerful system for acquiring it when it is scarce. But could that fact actually be turned into an evolutionary trap? In other words, when you attack a system that is essential to an organism, okay, it can't really easily change that without tremendous losses in fitness relative to other bacteria. So we designed an experiment to test this question. We also wanted to know whether the genomic background of the organism, in this case, silver resistance, did it influence how bacteria respond to excess iron 2, excess iron 3, or magnetite? Now, I'm not going to talk about the epistasis work in today's talk, but the principle here is very similar to a paper that we just published with the Lenski Laboratory at Michigan State in the Proceedings of the National Academy of Sciences, in which Richard's graduate student, Kyle Card, now Dr. Kyle Card, demonstrated that with regard to antibiotic resistance, genomic background played a major role in the capacity of the bacteria to evolve resistance to specific antibiotics. And so I reasoned in the same way that the genomic background, metal background, should impact how an organism responds and evolves resistance to a new metal. Now, my theory, and I think maybe naively so, was that because silver and iron have such similar mechanisms of toxicity, that an organism having a already existent silver resistance mechanism might give it an advantage relative to organisms that have no history of silver resistance. Now, that's another story for another day. So maybe I can come give that talk to you guys next year when I write the epistasis papers, which we finished all these studies, but the results have not been written and published yet. Now, to do the experimental evolution to generate iron-resistant populations, in this case, again, in this study, we're talking only about the iron 2 populations. We first determined an initial minimal inhibitory concentration. Now, to do an experimental evolution project, you obviously cannot try to grow the bacteria at their MIC. In other words, if you're at the MIC, none of them survive, none of them replicate. So you have to use a sub-MIC concentration. And the degree to which you use a sub-MIC will determine the rate at which the populations respond to evolution. So you don't want to use something that's too close to MIC, because that is going to lead to the potential of the population going extinct. Neither do you want to use something that's so far away from the MIC, because then there's very little or very weak selection, meaning the response is going to take a very long time. So we tried to find something that left about 15% to 10% of the bacteria surviving in the presence of excess iron. And for iron 2, that turned out to be okay, where are we at for iron 2? About 3,500 milligrams per liter of iron 2. And this was iron 2 sulfate. Now, here are the results of this experimental evolution. Now, we had evidence of iron resistance as early as 50 days. And because those cultures were contaminated, we could never publish that data. So we went on with the experiment in uncontaminated cultures to 200 days. And these are the results of that work. Now, the blue squares represent those that were selected for excess iron 2 resistance. The red circles represent the controls. The controls are grown in standard Davis minimal broth with no excess iron. There's a minimal amount of iron in Davis minimal broth, so the bacteria aren't starving for iron. So this is different from the ancestral population, which is taken directly from the sample of E. coli MG E. coli K12 MG 1655 that we received from the company. So these are stored in -80. We bring them out, we let them grow for 24 hours, and then we assay them. So they're as close to that ancestral physiological state and genome as possible. The controls grow for the same number of generations as the iron 2 selected, but in the absence of excess iron. So what we see from these results is that as we increase the concentration of iron 2, the iron 2 selected populations show superior 24-hour growth at all concentrations. The controls do slightly better than the ancestors, as we would expect, since the controls really aren't facing iron stress. When we compare them in iron 3, which is panel B, we see a similar result in which the iron 2 resistant population again shows superior growth in iron 3 compared to the controls. And the controls and ancestors are not statistically significantly different from each other. Now, when we compare them in gallium 3, now gallium is an iron analog. It has almost the same atomic nucleus radius as iron does. And so gallium has been proposed as an antimicrobial with the idea that you could quench iron siderophores because they won't be able to tell the difference between gallium and iron. Now, we've done experiments demonstrating that bacteria can evolve resistance to gallium as well, but again, I'm not talking about those results here. But what we see interestingly in this experiment is that iron 2 resistance also powerfully provides gallium resistance relative to the controls and the ancestor. Now, what we found shocking, and I must say I was floored when I first saw these results, is that iron 2 resistance confers no resistance to silver. The reason you can't see anything in this graph is because ionic silver kills the iron 2 bacteria, it kills the controls, it kills the ancestors, okay, at all concentrations tested. Now, we know that silver is one of the most toxic metal ions to bacteria, but I had actually thought that since the mechanisms of iron 2 and iron 3 resistance are so similar, or toxicity, excuse me, mechanism of iron 2 and iron 3 toxicity to bacteria and those of silver are so similar that selection for iron 2 resistance would also confer silver resistance. Turns out it doesn't. Now, to demonstrate that what we were really looking at was a response to silver, response to iron 2 and not the sulfate ion, we tested 24-hour growth in sulfate, and we found that the ancestor actually does better than both the selected population and the controls, which illustrates that the change that occurred in the controls and in the iron 2 selected population was to iron, not to sulfate. Now, another interesting result of these experiments that again could not have been predicted from the initial proposition was that antibiotic resistance was also a correlated trait with iron 2 resistance. So the iron 2 resistant populations became better at growing in ampicillin, better in growing in bacitracin, better at growing in polymyxin, better at growing in rifampicin. There was no difference in tetracycline, strong difference in chloramphenicol, and a strong difference in sulfanilamide. Now, the reason this result is important is because people who are doing material science research and attempting to deploy iron nanoparticles to control, multi-drug resistant bacteria aren't realizing that these kinds of pleiotropic correlated responses to resistance to iron occur. Now, we've also shown in a paper that's under review right now in nanomaterials using magnetite nanoparticles. Magnetite is a composition of both iron 2 and iron 3, that the same thing happens. So when we produce magnetite resistant bacteria, okay, they become resistant to iron 2, iron 3, gallium, okay, they have some minor silver resistance, and also all of the same antibiotic resistances that we show here occur in magnetite resistance. So this was, again, an unanticipated result of these experiments. Now, we also wanted to know the genomic foundations of iron resistance. So we extracted from each replicate population and prepared genomic libraries the depth of coverage between 30 to 40x for all samples. We called genomic variants via the BRESeq 30.1 pipeline, the methods of which are described in Dethridge and Barrick, 2014. Jeffrey Barrick is a colleague of mine from the Beacon STC, and we've done research together. He was a co-author on our first silver nanoparticle paper. So I have high confidence in the validity of this pipeline. So we filtered out all the ancestral variants before comparing population replicates. So for those of you who have done genomic research where you're doing resequencing, and I'm sure you're aware of this, but if you're not, I just want to be sure that you are, that the reference genome for these organisms that exists in NCBI and the actual organisms you receive from the biological supply company that sends you your bacteria are not the same. And that's because evolution always occurs. So whoever sends you your bacteria, they had to culture those organisms before they sent them to you. And in culturing them, they acquire mutations. Okay, so you must first sequence your ancestor and compare it to the reference genome. And then you're going to find that there are going to be some selective sweeps in things you would never have predicted. And you need to filter those out before you run your pipeline. Otherwise, you're going to be calling variants that really have nothing to do with your experiment. So we identified several variants that were not detectable in the ancestral population. And we report those here. So as you can see from here, we describe the gene in question, the specific mutation in it, and how it changed the protein in question, its frequency in the 5 selected populations. Now, we have a similar table for the controls, and what you find is that there's no overlap between the mutations that went through selective sweeps in the iron 2 populations and the controls. So that's why I'm not bothering to show them. Now, what the controls are adapting to is better growth in Davis minimal broth. And again, this always happens. Now, sometimes, and well, I'll tell you that when we see this specific mutation, but what we find is that there are variants that arise in systems that clearly have a relationship to dealing with iron toxicity. So this gene MERC, get the wrong way, this gene MERC is involved in cell wall formation and also in protection against ROS. QR is a copper responsive regulon transcriptional regulator that is involved in iron metabolism as well as copper metabolism. MERD-A, not really sure what this guy does, which is also a reason why we do whole genome sequencing, because sometimes we uncover genes whose relationship to a specific stress you would never have predicted. Okay, this gene MIF-A is involved in multi-drug efflux system protein. YAG is a protein kinase. with an endogenous substrate that's unidentified, but this was one of the strongest selective sweeps we saw. So clearly this gene has some impact on creating iron 2 resistance. Now, we also found mutations in PIPS-P, which is a component of the PEP-dependent nitrogen metabolic phosphoryltransferase system. Okay, but the most significant mutations we found are in FEC-A. FEC-A is iron 3 dicitrate siderophore transport system. So we found that each one of these populations, except for the fourth one. Now I can tell you that the fourth one did have a FEC-A mutation at generation day 50, but it was lost by day 200. So these have selective sweeps in the FEC-A gene. Now, we did computational modeling of these mutations to show how they interacted with the protein, demonstrating that these mutations actually made iron 2 binding to FEC-A weaker. So in other words, the way that these bacteria are surviving excess iron is actually by taking up less iron than they possibly could. Now, to demonstrate this further, we utilize a new technique for gene expression by a company named NanoString. And their technology allowed us to target 50 genes that we knew were involved in iron metabolism. Okay. And we were able to show that there was a clear differential in the presence of iron 2 in the expression of these genes associated with iron metabolism between, on the left-hand side, the controls, okay, who evolve in a condition without excess iron, and in the iron 2 population that evolve in a condition of excess iron. And so what the iron 2 populations are doing is they're down-regulating these genes involved in iron transport and uptake, which means they can essentially live in an environment with the amount of iron they need without killing themselves. Whereas the controls, when they go into this excess iron environment, are doing what they would normally do, and therefore are taking up more iron than they need, and this amount of excess iron becomes toxic. So our work was supported by NSF grant number 1602593 called Characterizing the Evolutionary Behavior of Bacteria in the Presence of Iron Nanoparticles. Also from the Beacon STC award, Using Evolution to Explore Antimicrobial Resistance to Spur New Antimicrobial Development. So with that, I will end my presentation and be happy to take any questions from the audience.
All right, thank you very much, Dr. Graves, for that very interesting talk. Now we open the floor for questions. If any of you have questions, so just unmute yourself, since we don't have many people, so just unmute yourself and then ask the question.
Dr. Graves.
Yes, go ahead.
This is Brian Vareli. I'm here at Virginia Commonwealth University. Pleasure to see you. I want to introduce myself and say that I was a PhD graduate student in Walter Eanes's lab at Stony Brook.
Back
in 1994, working on, I know an organism that was near and dear to you for a long time. It was inspired a lot by the work that you worked on and went on. I'll say that I'm no longer working on Drosophila and have moved on to human population genetics and plant population genetics and a lot of disease-related traits, and made a career out of moving all over the place, having a common theme of thinking about natural selection and evolutionary genetics. My question for you, and I share these things with all my students in all my classes, and I apologize that I missed the first 15 or so minutes here trying to get registered on Zoom, but one of the things I share with a lot of my classes and talking to them is you were the first African-American individual to receive a PhD in evolutionary biology.
Correct.
Correct. And what I would love to hear from you is, thinking about moving into so many different directions of research and having common underlying themes. What I think would be great to hear from you today is how you feel your background has brought unique perspectives to the field that you work in.
Okay, well, thank you for that question. Well, obviously, the place where my background most impacted this field was on the questions of interpreting human biological variation. Now, you undoubtedly know about the two books I've written that sort of summarize what an evolutionary perspective teaches you about human biological variation. And that's The Emperor's New Clothes, which came out in 2001 originally, and then was updated in 2005. And The Race Myth, Why We Pretend Race Exists in America. Now, I've continued to work on that question, particularly from the lens of evolutionary medicine, to try to update people's thinking about the nature of biological variation in our species and its association with complex predispositions such as disease. And you are, I'm sure, are aware that the situation is dire with regard to what medical practitioners and biomedical researchers think about human biological variation and how it plays a role in disease. So the good news is that Elsevier has retained me as a consultant because they're going through all of their medical books to address how they've been dealing with human biological variation and the confusion between socially defined racial groups and real biological entities. And so I've been working with a colleague in the pathology department at Duke, at Andrea Dreop, and we're right now revising the most widely selling textbook in pathology, Robbins Pathology, from the point of view of understanding how evolution works and how it plays a role in predisposing individuals to disease, and how none of that is associated with socially defined racial categories. So the bottom line is that's where my background has had the biggest impact in terms of how I understand and utilize evolution. And to give you guys a preview, all of that is going into my, well, actually two books. The first of those is coming out in the fall from Columbia University Press. It's called Race and Racism, Answers to Frequently Asked Questions. It has an entire chapter on, again, biomedical research and clinical practice. And the next book coming after that is called Voice in the Wilderness, How Evolution Helps Us Solve Our Biggest Problems. And that's going to be published by Basic Books, but that's coming out in 2022. So thank you for that question.
It almost sounded like my question was planted. It absolutely wasn't. That was an excellent answer. Thank you so much for that. I bring that up because of what's going on even right now, and this continues to consistently go on. It's not a new problem; it's an old problem that needs the same sort of solution, and that is with our pandemic going on, and how much of that information is really needed to infuse into our graduate students and having them think about how that's a problem. I recently reviewed a paper for New England Journal of Medicine where another reviewer who is anonymous, another reviewer writing back and saying, we really have nothing new to learn from people of color on topics of genetics. And it's just something that we look around and see what's going on right now in our culture with both social injustice and with our pandemic, and how they are obviously linked. And having individuals who are still reviewing literature, it's great to hear. I love that you're doing something that is literally at the foundation, going back and making sure that the literature is accurate. Thank you for your insight on that.
Anyone else?
Hello, this is Zhong. I'm a friend from Xianfa, and I'm a faculty member in VCU, also in School of Medicinal Chemistry. I'm a structural biologist, so your talk is very interesting, actually. I found through the adaptive evolution, you find some specific mutation in the transporters. So I'm wondering, is that kind of mutation is randomly happen, or because of the stress of the toxic ions?
Okay, thank you for that question. So mutations always occur. And this is, again, one of the things that, folks need to understand about, how variation happens. And that's simply because you can't replicate DNA without the, DNA polymerases and RNA polymerases making mistakes, right? So they make mistakes. And those mistakes then are exposed to natural selection. What's really interesting about these kinds of mutations, I've run them through BLAST, okay? They don't exist in nature. And the reason they don't exist in nature is for an obvious reason. You can't downregulate your iron, sequestration systems in nature and survive. Okay, so we don't see these mutations in nature at all. The only time we see them is in these excess iron environments that we've created in the laboratory. And when we take these bacteria and put them in standard Davis minimal medium, within several generations, not that many, those mutations disappear because they are a tremendous disadvantage. In Davis minimal broth is actually a scarce iron environment. So they have no capacity to compete with the ancestor or the controls who have adapted to sequestering iron in this low in iron environment. So the bottom line is mutations always occur, but natural selection acts on favorable genetic variants in specific environments.
Okay, good.
Okay.
All right, any other question?
Yeah, I have a question.
Hey, Martin.
Yeah, hi. I'm Martin. I'm from UVA. I work on microbes as well. So I have a question about the dynamics of the resistance. So in your 200 days experiment, does the iron resistance happen gradually or suddenly, in a rapid jump fashion?
We don't have measurements on enough generations to tell you whether it happened rapidly. The evidence would suggest that it occurred gradually, but again, it comes down to the acquisition of a specific variant and then the action of natural selection on that variant in that environment. So if I were going to guess based upon the data we have, by day 51, we showed clearly that these bacteria were iron resistant. They were growing better in excess iron than the controls could. So I would say that this was gradual because even after, when we go from day 51 to day 200, their growth in iron 2, excess iron 2, isn't dramatically different than it was in excess iron 2 at day 51.
Yeah, but okay, so you I guess don't measure them on day or every week, right?
No, we did not sequence every day, so I can't speak to that.
Not not sequencing, but measure their resistance.
Okay, so again, in terms of the measurements we have of their 24-hour growth, that indicated that they were gradually increasing their iron 2 resistance compared to the controls. Yes.
All right, thank you.
All right, any other question?
Yes, I don't know if I missed this, and it was probably at the beginning, but what is the natural environment of excess iron that these bacteria are being exposed to?
Okay, so in nature, generally excess iron does not exist. Okay, now there are some places, for example, iron ore deposits where the leaching of iron out of those iron ore deposits might create an environment where there's excess iron. So we know from other metal resistances that there are places in nature where you find bacteria that are resistant to excess iron, to excess silver, to excess copper, but all of those are in places where that metal is leaching. There's even bacteria that are resistant to excess uranium, but it's leaching from a geological deposit or a mine or industrial pollution. But in most, if we were talking about pristine environments, there is no excess iron because every microbe in there is fighting to get iron. And so that the excess just doesn't exist in places where you're not exposed to, anthropogenic increases in metal.
So do we come in contact with this bacteria at all?
You won't come in contact with these unless you come into my lab.
Right, in nature, right.
Right, but for example, let's take silver resistance. Okay, the first silver resistant microbes were isolated from a burn ward. And the patients there were being treated with silver containing creams. And pretty soon, okay, people found that the creams weren't working anymore. And they had iron resistant salmonella. I mean, sorry, not iron, but silver resistant salmonella. So the bottom line is where you use excess metal, just like where you use antibiotics, bacteria figure out mechanisms to become resistant.
All right, thanks. That put a more natural, bent on it. Yeah, thanks.
All right.
All right. Thank you, Jason, for the question. And any other question? I have one, Dr. Griffiths.
No, go ahead.
So you mentioned that the iron-2 resistance is positively correlated with antibiotic resistance. Do you have any hypothesis on that?
Yeah. So, basically, bacteria have like 5 standard mechanisms they use to resist poisonous things. So the first of those is prevention of entry, and that occurs at the cell wall, cell membrane. The second of those is efflux, meaning once that poisonous thing gets into the bacterium, they are, grabbed by transporters and then effluxed out of the bacterium. The third thing is alteration of substrates that are impacted by the poison. So one of the mutations that we standardly see in these metal-resistant bacteria are in the RNA polymerases, okay, either in the A alpha subunit, the beta subunit, the beta prime subunit, which is called RPOC. So we've commonly seen those. And to give you an idea of pleiotropy involved with RPOB, it's well known that mutations in RPOB impact the efficacy of rifampicin because rifampicin attacks the RNA polymerase subunits. So if we have a metal-induced mutation in RPOB, then it induces rifampicin resistance. And we found that in virtually every one of these RPO mutations that we've done. And by the way, we found these in silver resistance, in iron resistance, in copper resistance, in magnetite resistance, and every one of those shows strong resistance to rifampicin. Now, we've also found RPO mutations in bacteria growing in standard Davis minimal medium because RPO mutations in general also help with dealing with low levels of carbon source. And when you find one of those RPOB mutations resulting in the controls, they are also resistant to rifampicin. So the simple answer to your question is that some of these mutations are pleiotropic, meaning they impact several systems and therefore impact both metal and antibiotic. And there's evidence, again, from nature, you know, for the last question, that this co-selection of metal and metal and antibiotic resistance occurs all the time. So
Okay.
many of these mutations, you know, again, are multiply pleiotropic with regard to mechanisms associated with antibiotic action.
Mhm. All right. Okay. Thank you. Any other new question? Any other question? Well, in the last few minutes, maybe I would like you to share with us your personal experience in terms of what attracts you to the study of biological evolution, and what do you think we should do to attract more minority students, particularly African American students, into the study of evolution biology? I think that's
Okay, so that's, you know, that's a great question. So what I'm going to do is stop sharing my screen so people can see me better. So that's a really good question, and I addressed those in a paper that was published in Evolution, Education, and Outreach, called African Americans in Evolutionary Biology: Where We've Been and What's Next. And the good news is that compared to when I was a young researcher back in the late 80s, there's been a transformation of scholars of, you know, African descent in evolutionary science. So there's a new generation of very, you know, good researchers at prestigious institutions like Brandon Ogbuno, who's a professor at Yale now, Paul Turner, who was a professor at a student at UC Irvine. And quite frankly, if I hadn't been on the admissions committee was going to turn him away until I argued forcibly that they needed to accept him. Well, now Paul Turner is a full professor at Yale University, and 2 years ago was elected to the National Academy of Sciences. So things are changing with regard to who's interested. I originally got interested in evolutionary biology because I like to answer big questions. That's why I got interested in science to begin with. And as my lineal descendant, Thaddeus Dobzhansky, ancestor, I'm his descendant, he's the ancestor. So my lineal ancestor, Thaddeus Dobzhansky, said, nothing in biology makes sense save in the light of evolution. So virtually every biological research project is made more understandable if you put it in an evolutionary context. And in particular, you know, things that were spoken about by Brian with regard to disease predisposition, if you're not looking at that from this perspective of evolution, you just don't get it. So I was actually one of the founders of the International Society for Evolution Medicine and Public Health. And I invite all of you who are interested in doing, you know, or are doing biomedical research to join our society. The next Congress is going to be virtual, but we've already got a great lineup of speakers. I'm running a session on evolution and health disparity. So I would very much, you know, invite you to attend that. And if you like international meetings, the next meeting is scheduled to be in Lisbon, Portugal. So there's an opportunity for you in hopefully 2022 or 2023, depending on the progress of COVID. So to make a short answer to your question, evolution is important. It's fun. One other thing that I want to explain to people that's a common misconception is that you can be an evolutionary biologist and be a person of faith too. I am a confirmed Episcopalian. I attend church every Sunday. I work on the racial justice and social justice ministries of my church. I'm on the COVID vaccination task force of the Episcopal Diocese of North Carolina. So, you know, this is not a case of saying, oh, I can be a scientist or I can be a religious person, that's just wrong. Okay, you can do evolutionary science and go to church too.
How can we convey that message to, I mean, all the students particularly?
Well, one of the things you guys are doing was inviting me to give the seminar. That's a good thing. But what I'm going to do is I'm going to give you the names of, you know, this young cadre of African Americans in evolutionary science, and you should invite them to give seminars too. because
All right, yeah.
you need to see these role models.
Yeah, we can put that in the future seminars. Yeah, I think it's very important. Well, whether it's a task, you consider it a task or a mission or whatever. I think it's very important to attract more minority students in the study of African biology. And again, hopefully, many of them will be successful and set a new model for the other people to join the field. And not just the evolution biology, but also ecology as well. So yeah, so I really appreciate, and again, I believe I did the right thing to invite you to start the seminar this semester, and I appreciate you. I mean, give us this seminar and also all the talks we have today.
Again,
So,
great honor to have been invited, and I wish you all well.
thank you. Any other
Thank you,
questions?
Dr. Grace.
So, any other question or comment for Dr. Graves? If not, let's thank Dr. Graves again for the wonderful talk. And also, thank you for all the questions, very good questions, right? So again, our seminar will continue next week. Let me pull out the flyer just one more time for next week. I'll share with you on my screen. Okay. Dr. Sharhoff from Virginia Tech will give a seminar next week, all right, next Friday. So I'm sure it will be a wonderful talk as well, all right? So I look forward to seeing all of you next Friday. And also please spread the message to the other people you think who may be interested in the seminars, right? Okay, other than that, again, I appreciate all of you being here today. It's wonderful to have all of you to have for the first seminar. And I look forward to continue seeing you in the future through the semester. And for all the friends out there, wish you well. Hope you, well, you cannot see me right now. But okay. But again, for all the friends out there and all the participants, so wish you well. Hope you have been well. And I'll see you another time.
Okay, thank you. Bye.
Bye now.